Welcome. In this podcast, we'll learn about three types of chromophores and discuss how their light-absorbing properties relate to their UV visible spectra. By the end of this podcast, you should be able to, 1. Define a chromophore and recognize common functional groups and classes of compounds that are good UV-vis chromophores. 2. Name three types of chromophores and explain what characterizes each type. And 3, be able to predict the color of a compound from its UV visible spectrum, and be able to predict the UV visible spectrum for a compound knowing its color. Let's get started. As you'll recall, absorption of UV visible light by a molecule occurs when the energy of the light corresponds to the energy gap between the HOMO and the LUMO, and it produces a change in the electronic configuration of the molecule. In a molecule, there's a manifold of vibrational and rotational levels associated with each electronic energy level, which produces a spread in the energies associated with each electronic energy state. As a consequence, there is actually a narrow range of energies of light that a molecule will actually absorb. And as a consequence, when we shine white light, light representing all of the UV and visible wavelengths, and we record a plot of the absorbance as a function of the wavelength of light, which we call a spectrum, we see a band as depicted at far right. That's called a UV visible band. Excuse me. Recall, we can relate the absorbance of a sample to its concentration through the Beer-Lambert law, which says the absorbance at wavelength lambda is given by the product of epsilon, the molar absorptivity of the analyte at wavelength lambda, times the sample path length L, usually 1 centimeter, and the analyte's molarity in solution. A equals epsilon CL. Generally, we measure experimentally the absorbance at the peak of the band. We usually call the wavelength corresponding to this absorbance value the lambda max, or the wavelength of maximal absorbance for the compound. And I've highlighted this point in red on the spectrum shown at right. If you recall, bonding orbitals are lowest in energy, antibonding are highest in energy. Non-bonding orbitals are generally somewhere in the middle. So on this plot, I'm trying to show you the general pattern in terms of energetics for molecular orbitals. And note that the highest energy transitions should be sigma, sigma star, followed by pi, pi star, and then n pi star transitions, and it's based on the size of that energy gap. Again, highest energy transitions should be sigma, sigma star, followed by pi, pi star, and then n pi star transitions. So that means that the highest energy transitions, sigma, sigma star, should be at the highest energy or in the deep ultraviolet spectral region. Many peaks that are useful for UV-vis spectroscopic work are generated by certain functional groups that are really efficient at absorbing UV visible light. And we call these moieties chromophores. And on the next several slides, we're going to look at 4 major classes of chromophores. Our focus will be on understanding where we are most likely to see absorption occur in the UV visible spectral range for these compounds, and know something about how intense these absorption bands will be. In other words, how large the molar absorptivity values are. And recall that the molar absorptivity value for a material is a constant at a specific wavelength. It's a characteristic of the analyte. Chromophores are generally named based on the electronic energy levels involved in the electronic transition, responsible for the absorption of the UV visible light. And so generally they're given the name X-Y, where X represents the name of the highest occupied molecular orbital, and Y corresponds to the name of the lowest unoccupied molecular orbital involved in the absorption transition. The first two classes of chromophores we'll look at are the pi-pi star chromophores and the n-pi star chromophores. The highest occupied molecular orbital in a pi star transition is a pi orbital, and the lowest unoccupied molecular orbital is an antibonding pi orbital. The molar absorptivity associated with these transitions is very high, circa 10 to the fourth per molar per centimeter. And you typically see these transitions in the UV spectral range. This slide shows how UV-vis was used to identify artificial sugar substitutes. Aspartame is a dipeptide consisting of aspartic acid coupled to phenylalanine. Phenylalanine and saccharin, two popular sugar substitutes, both contain aromatic rings. The aromatic ring is a large pi star chromophore, and as such, it gives rise to absorption of UV light. And it turns out it absorbs light, as shown at right, in the UV spectral range near 240 nanometers. Cyclamate, saccharin, asulfine, potassium contain a sulfur double bonded to two oxygens. This is an n-pi star chromophore and absorbs at shorter wavelength. So let's return. So two types of important chromophores in the deep UV, pi star chromophores and n-pi star chromophores. Example of pi star, anything that's aromatic, anything involving double bonds. The other type is n-pi star. N corresponds to a non-bonding orbital, transition from that as the HOMO to a pi star as the LUMO orbital. And a good example of a chromophore that exhibits this type of transition is the ketone functionality and therefore acetone. Again, strong absorbance bands in the deep UV. Now let's look at the effect of conjugation on pi star transitions. If we looked at ethylene, carbon double bonded to carbon filled out with hydrogens, there's an optical transition that's between the pi star orbitals in the deep UV. It turns out about 180 to 200 nanometers, and that's bordering on the vacuum UV spectral range. The molar absorptivity for this particular transition is, again, about 10 to the fourth per molar per centimeter. Well, let's consider what happens if we add more and more carbon atoms into the system. As we do so, then the energy gap between the pi and the pi star orbitals begins to decrease because we have more pi and pi star orbitals because we're contributing more and more of these carbon-carbon double bonds to creating these molecular orbitals. And that means that the net effect is to decrease the energy gap as we increase the number of double bonds. By the time you hit hexatriene, the lambda max ends up being as far out as about 260 nanometers. There's also a secondary effect, which is equally important. As you increase the number of double bonds, there's a concomitant change in the molar absorptivity, epsilon, and it increases to be about 35,000 by the time you have this hexatriene system. We have the alternating double single bonds. One of my favorite examples of the effect of increasing pi conjugation are these carotenoids, which are responsible for the brilliant color of carrots and tomatoes. And on this slide, shown at right, are the structures for beta-carotene at top and lycopene at the bottom. Beta-carotene absorbs strongly in the visible spectral range between 400 and 500 nanometers, and lycopene, which has two additional conjugated double bonds, absorbs strongly between 420 and 520 nanometers. The absorbance for lycopene is red-shifted due to the extended delocalization because we have these additional conjugated double bonds in the main chain of its structure. Pretty cool. Here's another example of an important chromophore found in nature. many proteins and enzymes are based on a heme structure. A heme is a porphyrin, and the porphyrin is what's depicted on the slide, into which an iron inserts itself in the center. The iron binds to the 4 central nitrogens. Hemes tend to form octahedral geometries, and this comes out of a discussion of ligand field theory. And the axial ligands that it binds in proteins are amino acid residues from the polypeptide chain. And that polypeptide chain usually wraps itself around the heme group. So the heme group is generally buried in the center. The pi star transitions of this extensively delocalized heteroaromatic ring system, and I want you to take note of it. This is an extensively delocalized heteroaromatic ring system. They produce the distinctive red color of blood and the characteristic ruddy red of muscle tissue. Again, pretty cool. Well, there are other kinds of chromophores, so I want to talk for a moment about 2 more important classes of chromophores, and these are only going to be found in compounds that are based on transition metals. And these transitions are referred to as charge transfer transitions and ligand field transitions. Charge transfer transitions are exactly as the name suggests. The electron or the charge moves between 2 energy levels whose character is quite different. There are 2 flavors of charge transfer transitions: metal to ligand charge transfer, or MLCT transitions, and ligand to metal charge transfer, or LMCT transitions. In MLCT transitions, again, metal to ligand charge transfer transitions, the highest occupied molecular orbital is an electronic orbital localized primarily on the central metal. Hence, it's M as the first characteristic in this transition. And the LUMO in these transitions, the lowest unoccupied molecular orbital, the orbital where the electron will be transferred to, is an electronic orbital primarily centered on the ligand. And a ligand is the residue, the organic residue to which the metal binds in a transition metal complex. So in MLCT transitions, the HOMO is metal-centered, and the LUMO is ligand-centered. And I hope, therefore, it makes sense that in LMCT transitions, it's the converse. The HOMO is ligand-centered, and the LUMO is metal-centered. Charge transfer transitions are generally found in the visible spectral range, and they have molar absorptivities that are still significant, but they are lower than those of the pi star transitions, typically on the order of 100 to 1,000 per molar per centimeter. Excuse me. The last type of chromophore I'll discuss are ligand field transitions. These are transitions between 2 metal-centered orbitals. These bands appear in the near-infrared spectral region, so these are wavelengths generally further out than about 700, 800 nanometers, and typically these transitions have very low molar absorptivity values, about 1 per molar per centimeter. So let's take a look at a compound that has an MLCT transition. The compound shown on this side, ruthenium trisphenanthroline is its name. The ligand here is phenanthroline, and there are 3 of them. They form an octahedral complex around the ruthenium, which has a 2+ charge. So we call this ruthenium trisphenanthroline. And this is a really important compound. There are a series of compounds based on ruthenium binding to these heteroaromatic ligands. Another important compound is ruthenium tris-bipyridyl. The ring system in ruthenium tris-bipyridyl is a 2,2' bipyridyl, by the way. Ruthenium trisphenanthroline happens to have a metal to ligand charge transfer transition, and it goes from an orbital centered on the ruthenium to an orbital centered on the phenanthroline ring. And the band actually appears in the UV visible spectrum around 450 nanometers. Now, what's interesting about this compound is that it also emits light from a triplet state. So having said that, hopefully we're all on the same page. We're talking here about phosphorescence in terms of the emission. And the phosphorescence is centered around 610 nanometers. Of course, that's redshifted with respect to the UV visible band. And the emission of ruthenium trisphenanthroline and ruthenium trisbipyridyl has been used widely as an emissive tag on a variety of different materials in order to detect them. Now, we've already discussed this point earlier, but I want to remind you that associated with each of the electronic molecular orbitals, there's a manifold of vibrational and rotational sublevels. And so the consequence is we see bands when we absorb optical spectra for compounds. And this is as versus intense lines, which is what we would see if we were dealing with atomic elements. And that is the case when we end up eventually talking about atomic absorption spectroscopy, or AAS. On this slide, I want to show you a UV visible spectrum for a real compound. So this is a real measured UV visible spectrum. It's for a compound I hope you've actually seen in the lab, potassium permanganate. It's a potent oxidant. It's often used in general chemistry lab experiments in terms of redox chemistry. And what you're looking at is a UV visible spectrum across what's considered to be the standard UV visible range, 300 to about 800 nanometers. And this has been recorded for a 3.3 millimolar solution of potassium permanganate in a 1 centimeter cuvette. And I've also talked about this before, but I want to remind you, 2 is about the highest absorbance that you can measure in most instruments reliably. And I've got a couple peaks labeled here to give you a sense of the reality that for most compounds, you don't have just one single optical transition. There may be more than one. But what I want to focus on here today is the charge transfer transition. And I've marked for you that there are 2 overlapping transitions. There's actually a whole manifold of transitions centered around about 530 nanometers. And hopefully that says to you that we're talking about like a green color of light being absorbed. And so it should make sense to you, since all the other colors of light are going to be transmitted, that the mixture of those remaining colors is what we're seeing, and the compound is therefore purple. Pretty cool. So let's look at another compound now that we've just talked about. We've talked about hemes, and on this slide you're looking at the UV visible spectrum for a protein, cytochrome C. It has 1 heme with an iron 3 plus in the center. And you'll see that, again, there are multiple bands. And the 2 I want you to focus on are the band at 408. It's often referred to as the gamma band or the Sorey band, after the individual historically who discovered this important distinctive optical transition for heme systems. And then there's a pair of overlapping bands with very relatively weak intensity down centered around 528. These 2 bands, 408 and 528 nanometer bands, are both pi-pi star transitions. And in this case, most of the light that's being absorbed is going to be the blue light. And therefore, the result is that when we look at this compound in solution, what we see is an orange solution. Now, I also want you to take note here that I've recorded for you this UV visible spectrum for this heme protein at a concentration of 10 times 10 to the minus 6 molar. And that's to let you know how very intense this Sorey band is. Why is it so very intense? Molar absorptivity is actually about 100,000. It's because it's this extensively delocalized heteroaromatic system. Remember, the 2 main effects of this conjugation are a redshift in the pi-pi star transition and a concomitant increase in the molar absorptivity. Again, pretty cool. Well, I want to show you one more pretty neat thing. what happens if we add a reducing agent to this protein? And so if we add a reducing agent to ferri cytochrome C, that's what I'm showing you in this spectrum, are the optical changes that take place. If I look at the solution, it immediately changes from orange to a really pretty pink color. And what you're seeing are the distinctive changes in the UV visible spectrum that produce that change in color to this pink solution. So the heme Serre band shifts from about 406 nanometers out to 414 nanometers. And notice there's an increase and a separation in the two overlapping bands that I showed you earlier that were centered around 528 nanometers. Now they're well separated and significantly more intense. This is the result of a change in the oxidation state of the iron. And why are these changes happening? We've changed the electronic structure, and therefore we've changed the energies associated with the HOMO and the LUMO, and therefore the optical transitions that are involved. So another extremely important use of UV visible spectroscopy historically has actually been to learn a lot about chemical structure. I want to conclude the podcast by taking a look at several common biochemical analytes in proteins and DNA. And apologies for all the coughing. There are 20 common amino acids, which basically represent the building blocks for proteins. And on this slide, you're looking at 3 of these amino acids, which have aromatic and heteroaromatic rings. Because they have these aromatic rings, they have pi-pi star transitions, which, as we've said earlier, should give rise to intense transitions in the UV spectral range. And on this slide at right, you can see the UV visible spectra for these 3 aromatic amino acids, and I've displayed them one on top of one another. Tryptophan has what we call an indole ring system, an aromatic ring bound to a 5-membered a ring with nitrogen and an extra double bond, and that double bond is conjugated with the ring system. And for this reason, the absorption band for tryptophan is redshifted and significantly more intense than that for either tyrosine or phenylalanine. And the absorption wavelengths and intensity for tyrosine and phenylalanine are easily understood when you think about the structural differences. Tyrosine is basically phenylalanine with an electron withdrawing hydroxyl moiety attached. The electron density from the oxygen is coupled with the aromatic ring system, and this ends up producing a transition more intense and redshifted compared to phenylalanine. So it might not surprise you that a simple quantitative assay used for many years to quantitate protein concentrations is based on knowing the primary amino acid sequence of the protein and determining how many of these aromatic amino acids you actually had. And if you knew how many you had, the molar absorptivities were known for tryptophan and tyrosine and phenylalanine, you could basically consider this to be a mixture problem, which we talked about in class Thursday, where tyrosine and tryptophan were the primary contributors. And so basically what people did is they would sample the absorbance at around 280 nanometers and consider it as a mixture problem and solve. Pretty cool. Let's talk for a moment about DNA. On this slide, I'm showing you the bases that make up DNA. And if you'll recall, there's 4. 2 purines, adenine and guanine, and 2 pyrimidines, cytosine and thymine. And what I want you to focus on is the structure of these heterorings that make up the bases. And again, I point you to the fact they are pi-pi star chromophores. So they're going to produce intense UV signatures in the same spectral range, roughly, as for proteins. Because of the nitrogens, though, the transition is going to occur at slightly higher energy, and the bands are blue shifted compared to those of the amino acids. And so you can actually assay for DNA and do basically the same concept in terms of quantitation at around 265 nanometers. And with that, this will bring this podcast to a close. And before we stop, though, I'd like you to take a moment, pause the recording, and make sure that you can, one, define a chromophore and recognize the common functional groups and classes of compounds that are good UV-vis chromophores. Name the different types of chromophores and explain what characterizes each type. And lastly, be able to predict the color of a compound from its UV visible spectrum and be able to do the converse. Predict the UV visible spectrum for a compound if you know its color. And with that, I hope you have a great weekend.